Hello and welcome to this tutorial for Synteny. My name is Harry and I'm a PhD student in the David James Lab at the University of Sydney. My work uses proteomics to decipher how differences in protein expression between individuals are linked to metabolic diseases like type 2 diabetes, and I'll be with you throughout this tutorial. We built Synteny to help bridge the gap between studies in model systems and large-scale human genetic datasets, and hopefully by the end of this tutorial you'll have an understanding of how you can best apply this to your own research. So we'll start with the workflow of retrieving human genetic evidence stores. We'll use an example from a recently published paper to do that, and hopefully that's similar to a situation you would encounter with your data. Next, we'll use human phenotype enrichment to see whether this gene set is enriched for any human phenotypes, and we'll have a look at how you can interpret the results of that analysis. And then finally, we'll look at what is the phenotype tab for. And this tab is actually really useful for understanding how human phenotypes relate to each other. So the example that we'll use comes from this recent paper from the James Lab, where they looked at different adipocytes from mice from different adipose depots, so either visceral fat or subcutaneous. What they did was isolate the adipocytes from those depots from mice fed either a high-fat diet or a healthy chow diet, and then perform proteomics on them. And they saw something like this. They found that when you did the proteomics, you found approximately 6,000 proteins. There were many proteins that were upregulated and a similar number that were downregulated. And the question really is, which of these changes are causal? The reason this is an important question is that if you're designing therapies to treat metabolic disease or any complex condition, you want to target the ones that are actually causing the pathology rather than the ones that are downstream of that. And so in this example, because the pathology was induced by a high-fat diet, you can imagine that there are many responses to the high-fat diet in the proteome that don't reflect the causes of the pathology. And so we're really just interested in the causes. And to do that, we're going to need a data format that is a comma-separated variable, or CSV file. And that looks a little bit like this. So if you're using Excel, it would just be a list of gene names where there are no row names, no column names, just gene names. So very simple data format. And if you want the huge scores, you just need one of these files, so just the genes of interest. But if you want to perform human phenotype enrichment, it's also advisable that you have a second set of background genes. And the reason that these background genes are important is that if you perform your analysis from a tissue proteome or something like that, so in this case we've got adipose, you'd expect that you might detect enrichments for phenotypes that the adipose tissue is most involved in. So maybe lipid homeostasis, anything like that for adipose tissue. But we're not really interested in what the tissue does, we're interested in what the specific genes do. And so to correct for that tissue effect, it's necessary to provide some background genes to account for that. And so in the end, what we'll end up with is, relative to the tissue, what are these genes doing? Cool. So this is the Synteny browser. As you progress through the app, these tabs with data analysis and enrichment results will appear up the top here. So whenever you perform a new analysis, just see if there's a new tab. And so if we click this button to start, you see that this data tab has now appeared above. And so this is where we'll input our data. So we can begin doing that by uploading a gene set here. Browse for that CSV, and we'll just put our DE genes from the adipose tissue example. We open that, uploads. Down the bottom, it tells you that 473 genes have been uploaded, and that should just be the number of genes in your list. And then it's saying that the species that it's auto-detected is mouse. Now, if you had a different species, it would auto-detect that from any of these model organisms here. If it gets it wrong for whatever reason, because there's an overlap for mouse and rat, for instance, you can change that, and it would then change how it maps the orthologs back to human genomes. Go ahead and we'll acquire these huge scores. And whenever you do anything in Synteny that takes some amount of time, you'll see that there's these progress bars down in the bottom right-hand corner. These will just let you keep an eye on how you're performing the analysis. And so here we can see that it's also told us that of those 473 genes, 444 have been able to be mapped. What this means is that some of these genes may not have orthologs, or that we've just got the gene names wrong. So for the 30 or so that it can't map, it will produce those below here. We'll be able to see that and potentially correct them for future analysis. You can really see the speed advantage here in that if we were querying these genes one by one, it would take us hours to do this. You may never be able to do 400 genes manually. The power of that to unlock new discoveries is really quite impressive. So it's now finished acquiring all the information for probably more genes than anybody could study in one lifetime. Let's have a look at the results. As we talked about before, there were those 30-odd genes that it couldn't map to human orthologs. We can see that some of those are, because that's not a gene name, that's some uniprot ID from an uncharacterized protein, so not really surprising it couldn't map that. Some of these others, we'd have to look into specifically why there is no ortholog, but probably because there just isn't a human ortholog for that mouse gene. When you're looking at the gene sets of this size, it's not really surprising either. So once you check that, you can access your data via either one of these new tabs that has appeared. So huge for looking at huge scores and enrichment for performing human phenotype enrichment. So we'll check out the huge tab first. And here we'll see that there's this really big, beautiful, complicated heat map. This is fantastic. There's so much data here about how these genes relate to human phenotypes. But there's actually too much data. We can't see what's going on. Rows, they're the genes that we had in our gene set, and the columns, they're the human phenotypes. But we need to simplify this because it's uninterpretable. Probably the most common question we get asked about this tool is, what do the huge scores mean? They vary between 1 and many thousand. So how do I interpret them? So to get access to a guide for interpreting them, just scroll to the top of the page and click where it says here. Sort of 30 is a pretty good huge score. And so just to simplify this, we'll pick a threshold of 100. And so if we go down to this minimum score to plot, we pick 100. This will now only include genes that have an extreme huge score. So this is a very strong genetic evidence linking them to disease. And that simplified this a lot. So there's now only so many phenotypes we can actually read what these are. But it's still a bit hard to interpret. Still a few too many traits. So one option here is to lay this out by phenotype group so that we can see what types of phenotypes are associated with these genes. And you can do that here with the phenotype layout. So at the moment they're clustered. You see this kind of groups them together by how they're behaving. But if we do this by trait group, you can now see that it's grouped them by type of trait they are. So we've got all of the glycemic traits, the hematological, the hepatic, and so on. And these are just in alphabetical order. So if you know what you're looking for, quite easy to find. We've now simplified this to the point that we can start to look into some biology, and to narrow it down a bit further and look at the individual traits. We have to bear in mind, where did we get these data from? So we got them from adipose tissue, and so that would be, in this context, most related to obesity. So let's look for things that could be complications of obesity. So how about cardiovascular, glycemic, those types of traits? So if we just want to filter the heat map for those, you can just select specific phenotypes down here. And so if we remove all the phenotypes, we can now search for the group that we want. So we'll start with cardiovascular. If we click cardiovascular, you can see it's selected 73 traits that are within that cardiovascular option. Okay, so all complications of obesity, and all up that's 223 traits, but there's probably only 50 or so within this heat map with very high, huge scores. So to see what these are in a bit more detail, label the phenotypes. Yes. And the plot automatically adapts to now show you what the phenotypes are. When you do that, the label for the trait group goes away, but you get this colored box at the top, you get a little legend, and the numbering tells you from left to right. Just looking at this, we can see that most of these genes here that are enriched seem to be enriched for these lipid-related traits, in particular HDL, cholesterol, triglycerides, all things that adipose tissue might feasibly regulate. So the idea that the high-fat diet feeding is actually regulating these through changes to the adipose proteome is quite exciting. And so if we want to have a look at which genes in specific are contributing to this, it's possible to look at the raw data. So this is tab here. And to start with, let's just have a look at which genes contribute to HDL cholesterol, because that might be a phenotype of interest. And we can see that there's a list of genes here with very high huge scores. So remember, anything above 100, that's an extreme level of genetic evidence. This CD68 with almost 1,000 is an utterly gigantic huge score. If you were to click on this gene hyperlink, it would take you to the association to function portal, and you'd be able to learn more about the gene there. Conversely, you can always look for genes as well. So one of the top regulated genes in the adipose proteome was a gene called NPL. And so if we just want to look at what is the genetic evidence, or is there any genetic evidence for NPL, just search for it in much the same way. And we can now see that NPL has a giant, huge score for obesity. And so potentially the change in NPL that we see with high-fat diet feeding is actually partly causal for the obesity phenotype that we see in these mice as well. Okay, so this is the sort of main gist of this tab. And if you're happy with the plot at the end, you can download the heat map, and whatever form you see within the viewer, that will save as a PDF straight to your computer. Last thing to point out here is that if you want the raw data, you can download that here and it will download an Excel file for you. So next we'll move on to human phenotype enrichment. The reason for doing this is to ask, are any of the genes within this gene set enriched for causing any human phenotypes? And so we talked about background in the introduction. So here we're going to need genes that are the background of the adipose proteome. And that's really important so that we don't just detect spurious associations for adipose phenotypes. And that's in the same format as the gene lists. See, we've got all of the proteomes, almost 6,000 genes there. In terms of selecting which phenotypes you can test, you can just go through manually and pick phenotypes. You can search for this, so it's interactive. So if you wanted HbA1c, for example, you can search for that, anything like that. I would advise that you test all. That can be quite time-consuming, but it offers you the most possibility for discovering something interesting. For the purpose of this, though, we'll just test the phenotypes that were in our heat map on the huge page. And so those are the 52 phenotypes that had a huge score of above 100, so a extreme level of genetic evidence. So when you're ready to run the enrichment analysis, you can perform the enrichment. And again, you get a little progress bar telling you what the progress of this is. Fantastic. So that's finished too. See that it's telling us to scroll down to see the results. And if we do that, we're now greeted with this bar plot that shows us the results. It is showing us phenotypes that have an enrichment greater than 1. So that means anything which is somewhat enriched. If it's a purple bar, that means that the adjusted p-value, so the false discovery rate corrected p-value, is significant. And if it's got a star, the star corresponds to the unadjusted or nominal p-value. In terms of the settings for this plot, you put the background. So the default is just the human genome. So if we correct for that and use our background gene set, what we see is that there are now 2 phenotypes that are significant, both at the unadjusted level, but very significant phenotypes. This button here will also change the table below. So the table of results just reflects the bar plot. And so there are these 2 phenotypes here that are significant with respect to the adipose proteome. And so those are UPCR, urinary potassium to creatinine ratio. That's a kidney-related phenotype that is indicative of kidney damage. And then mean arterial pressure, which is also a very strong risk factor for kidney damage. And so potentially the insight from this is that the proteins that change in the adipose tissue following high-fat diet are actually causing increases in arterial pressure that then lead to early-stage kidney damage. Quite an exciting hypothesis because the relationship between obesity and kidney disease mechanistically is not very well understood. But this provides a way of identifying the genes that are doing this, and then encourages researchers to test them mechanistically, either in small animal models or cell models or what have you. But really, the purpose of Synteny is to encourage people to validate things mechanistically and to give them new and exciting hypotheses. So as with anything else on Synteny, anything that is highlighted in purple is a hyperlink. And so if you click on these phenotypes, they will take you to the association to function portal where you can learn more about those. Now, it's important with this enrichment to understand which genes are contributing because the huge scores are aggregated across the whole gene set. And so we want to know, is this phenotype enriched just because we've identified several very high scoring genes, or is that enrichment signal evenly distributed through the genome? So you can do that using this gene level contribution. For the phenotype that you're interested in, you would search for that. So if we go for mean arterial pressure as our phenotype, we can see that all of the red dots are the phenotypes
other genes that were in our gene set, and then all the other dots are all of the other genes in the genome ranked by huge score. And so you can see that most of our genes actually don't have a particularly strong huge score, but there are some that have very, very high huge scores. And so in the case of mean arterial pressure, that enrichment is probably driven by these genes rather than the rest. Moving on to the phenotypes tab, it'd be good to show you just how you can use this. So there are 3 tabs within this, and the first is just a reference of all the phenotypes and their IDs. So you may have noticed in the previous sections that Synteny reports the phenotype IDs. And so we found the urinary potassium creatinine ratios UPCR. If you didn't already know what that was, you could search for it and it would tell you. It would also tell you which group that phenotype sits in, and that would help provide a bit more context. If you're interested just in looking at what phenotypes are in the database and you have a particular category in mind, you can search for that. So if you wanted to know all the cancer phenotypes that the Association for Function database has, you could scroll through. You can see that here they have 19 different forms of cancer phenotypes. So they've got all forms of organs, many different cancers. And so this database is very impressive. It has almost over 1,000 phenotypes, and that can be used to provide a lot of insight for whatever biological context you are interested in. The second thing that this module does is give you access to genetic correlation data. So this data is all captured within the Association to Function portal. The user interface there is just a little bit challenging to use, and so we've made a module here to make that a bit more accessible and contained within the Synteny tool. And so the reason for using this is that often you will identify phenotypes that you haven't seen before. So the urinary potassium creatinine ratio is a good one of those. And to help you understand how those relate to other aspects of physiology, genetic correlations can be very helpful. So the genetic correlations tell you what is the extent of shared genetic cause for each trait with every other trait in the database. So if we just search for that trait now, and then we load the data, you can see that these are all the traits within the database, and the results of this are just graphed over here. And this is also interactive, so you can hover over the points. And so you can see that UPCR is correlated genetically with a bunch of kidney phenotypes, the sodium potassium ratio, sodium excretion, albumin to creatinine, which is a more well-established kidney health marker. But then it's also genetically correlated with weight, body mass index. And so these potentially is where that driving force from our obesity model is coming in, and that the genes that we're detecting are changing in response to weight and BMI, and they are driving this change in kidney function. And so there's a potentially very interesting hypothesis and set of relationships between diseases that we wouldn't otherwise have been aware of. And just to show you that this does work and that this is a reproducible and robust way of looking between phenotypes, let's say you didn't have any concept of how type 2 diabetes related to other aspects of health. So if we get this for type 2 diabetes, we can see that one of the top phenotypes for type 2 diabetes is HbA1c. And so it's saying that the genetic causes of a HbA1c almost completely overlap with the genetic causes of type 2 diabetes. And this is unsurprising because HbA1c is actually used to diagnose type 2 diabetes. It's a measure of how well your blood glucose levels are managed. This tells us that these genetic correlations really do provide quite a robust measure of how traits relate to each other. And it's completely unbiased as well. That brings us to the end of this tutorial for Synteny. I hope that this has helped you understand how best to use the tool with your own data. Just to wrap up what we've learned how to identify high priority genes. In our case, we found CD68 as well as a couple of others. And that's led to an interesting question that if we manipulated these therapeutically, would that be beneficial in the treatment of obesity in our case? But when you expand this to other data sets, there are many novel insights that can stem from this. And so this is a really interesting approach. Secondly, we found some enrichment for traits relating to kidney function. And this was quite an interesting hypothesis because the idea is that changes in the adipocytes following high-fat diet are leading to changes in protein expression, which are causal for kidney dysfunction. And so potentially this might help piece together how obesity and kidney dysfunction are related mechanistically.
Thank you for sticking to the end of this tutorial. I hope it's now clarified how the use of these large-scale human genetic data sets can supplement omics data and really be an important part of that bioinformatic toolkit. For us, it's the scale of Synteny that makes the most difference. And the more genes and the more phenotypes you analyze, the greater your potential insight into your existing data you can unlock. If there are any questions that you still have that weren't answered by this tutorial and aren't answered by the frequently asked questions section, you can scroll up to the top of the help page and there's a link to my email address. You send me an email with the problematic gene set or what you'd like to know. I'd be more than happy to do my best to help you out with that.